So good morning, everyone. I'm Kyowon Jeong from Seoul National University in Eric Kim's lab, and I'm really thrilled to present my work on multiplex protein quantification today. But before I start, I'd like to thank the organizer for giving me such a great opportunity. So my work is about the protein quantification via LC-MS-MS, but because I do not think lots of audiences are familiar with LC-MS-MS, I will take a moment and briefly explain LC-MS. So MS is nothing but a very expensive scale that measures the weight of molecule instead of the meat, like this. So in proteomics, in LC step, the peptides are chemically separated. But in some cases, they may coalesce, as in this example. So in the following MS step, they are again separated by their masses, more exactly their MZ values, mass to charge ratios, as in this example. So they are separated. And the signal from each peptide is called XIC, the extracted ion chromatogram. Then the ions within a small point in this XIC is isolated and fragmented. And in the following, the second MS step, their fragmented masses are measured, which is used to identify the peptide by using existing great algorithms. So the ID is therefore assigned to the point on this XIC. So back to the quantification. I will focus on the isotopic labeling-based quantification, and because my tool is about the isotopic labeling-based quantification analysis. And in isotopic labeling-based quantification, the peptides, the same peptides with different conditions, are labeled by labels with almost identical chemical properties, but with different masses. So this is an example from SILAC, widely used metabolic isotopic labeling. The labeling masses are 0 Dalton and 6 Dalton. And they are labeled, mixed, and analyzed in the same LC-MS-MS run. So the XIC from the green one will appear in here, for example, and the XIC from the red one will appear in the same retention time range, but 6 Dalton shifted MZ value from the green one, as in this example. So their heights or their areas are used for quantification, but note that due to natural isotope, the actual signal will look like this. So one of the hurdles to increasing multiplexity in isotopic labeling is that enough mass spacing should be secured. Otherwise, the signals from differently labeled ions will overlap. As in this example, they overlap and they are added up, causing bias in quantification. Another hurdle is the retention time shift due to deuteriums in labels. To have various mass shifts, in other words, to increase multiplexity, the use of many deuteriums is one option. But if you use a lot of deuteriums in labels, you will get this retention time shift marked by delta RT, like this, again causing the bias in quantification. So the use of many deuteriums is also limited. So for these reasons or others, predominant isotopic labeling schemes have up to 3 plexity. Only 3 conditions can be analyzed at the same time. But high multiplicity, in particular, the multiplicity higher than or equal to 6 can be really beneficial for lots of biological applications, such as time point study or detection of differently expressed proteins using test condition triplicates. Also, increased multiplicity, for sure, reduces the statistical or experimental efforts. So people already tried to increase multiplicity for isotopic labeling, but most of them were not very successful. On the other hand, there are isobaric labeling schemes such as TMT or iTrack achieving high multiplexity. However, they have a major drawback, the compromised quantity ratios and quite a narrow dynamic range. So Stephen Gigi group reported this problem and suggested a solution to use MS3 step to mitigate this problem. But MS step is not available for many mass spectrometers, and it also reduces the number of identification. So in this talk, I introduce EPIQ. It stands EPIQ Protein Integrative Quantification, the software tool that enables n-plexed isotopic labeling-based quantification. EPIQ allows mass spacing as small as 2 Dalton and dozens of deuteriums in the label. I will also demonstrate that EPIQ has, EPIQ has low LLOQ, the lower limit of quantification as compared to gold standard quantification tool MaxQuant. And these are made possible by using the model-based reconstruction algorithm that I will describe in the next few slides. So I will introduce the model first. Our algorithm is based on the model that describes how the labeled peptides generate signal x-axis called x-axis cluster. I will show you the two example for the model for P1 and P2, two differently labeled the same peptide species with known quantities Q1 and Q2. So let the mass spacing is only 2 Dalton. We will see how this observed x-axis cluster is generated from those P1 and P2. So from P1, the model assumes the following 3 elements are known. The first one is the position of P1 x-axis in the MZ retention time plane. And the second is the shape of the x-axis. And the last one is the isotope distribution of P1. So by combining these elements, we can know the shape and the exact position of x-axis from P1, which is called template of P1. But note that template does not have any quantity information, but it only has shape and position information. So from P2, we can the model generate the template in the same way, and by finally multiplying the quantities into quantities to each template and superimposing them, we have the the model generate the x-axis cluster. Note that we also assume the presence of retention time shifting here, marked by delta RT in this example. So it was the model, and given the model and the observed x-axis cluster, we want to obtain the quantities. So before that, we first estimate the template per label, and the 3 elements, position, x-axis shape, and the isotope distribution are to be estimated. Assume the identification is given for this black dot in label 1 XIC. Actually, in practice, the ID can be assigned to label 1 or label 2 any label. So the ID gives this many information, and from the composite information, the isotope distribution is estimated using an efficient approximation algorithm. So 1 element is now estimated, and the next one is the position. First, the MZ position for label 1, the identified label is given from just the identification, like this. And because we know the mass spacing, the MG for other labels can be calculated very easily. But in case of retention time, position is a bit tricky. For the identified label, it is relatively easy though. Just find the closest apex in the XIC, and then it will give the correct retention time position for the label, for the identified label. This is because most often the identified label has a very high abundance, and thus its XIC shape is very clear. But for other labels, in many cases, the apex is very hard to be found. So instead of finding the position from the observation, we use the regression model to predict the retention time shifts between labels. I will skip the detail, but by using the predicted retention time shift delta RT, the positions for other labels can be estimated, like this. So now we have the second element, the position, specified by red and green dots here. So the last element is the x-axis shape, and it is found by fitting log-normal probability density function against the observed x-axis around the estimated positions. This is based on our observation, the x-axis shape fits well to the log-normal PDF of different parameters, like this and like this. So we have estimated templates. The weighted summation of these templates should constitute the observed x-axis cluster. The reconstruction is to find the weights Q1 and Q2 that minimize the error between observed and reconstructed ones. This is a well-formulated least square fitting problem that can be easily solved by using pseudo-inverse. And by solving this over-determined linear equation, the overlapped x-axis, overlapped signals are effectively deconvoluted. Now I'll show you some results. We designed diethylation 6-plex labeling scheme to test EPIQ and prepared Hela Lysate, very complex sample with input ratio of 30 to 20 to 5 to 1 to 5 to 10. So diethylation 6-plex labeling has at minimum 2 Dalton mass spacing, very narrow mass spacing, and up to dozens of deuteriums in label. So it's kind of very harsh situation. And using Q-effective, very widely used spectrometer, 16 fractionated data sets were generated and analyzed by EPIQ. EPIQ successfully reported about 5,000 all labeled quantified proteins. So proteins whose labels are all quantified. Sometimes it fails to quantify some labels if the quantity is just too small. And this box shows the log to fold change from the label 1. The dotted black line specifies the input ratio, so the reported ratio was not seriously biased for any label, as you can see from this red box. Okay. We also benchmarked EPIQ against the MaxQuant, and since MaxQuant can handle up to 3-plex, we prepared diethylation 3-plex Hela Lysate dataset with input ratio of 1 to 10 to 20, and the dataset was then analyzed by EPIQ and MaxQuant. This labeling has wide mass spacing of 4 Dalton, and it doesn't have any deuterium for MaxQuant, actually. So this is the result for overlapping peptides, not only more peptides quantified EPIQ, but also the ratio was less biased than MaxQuant. In particular, when low abundance peptides were examined, low abundance ones, the difference was even more explicit. This implies that EPIQ has lower LLOQ than MaxQuant. To check if this is really the case, we first counted the number of unquantified labels per label. While EPIQ has unquantified ones mostly for the label 1, the least abundant one, MaxQuant has many unquantified ones even for label 2. Also, as we know the input ratio, the unquantified label quantities can be estimated by using other quantified labels input ratio. And this shows the distributions of the estimated unquantified label quantities. The lower, the better. It is shown that the difference is almost 5 times, demonstrating EPIQ has about 5 times lower LLOQ than MaxQuant. So to sum up, EPIQ enables highly plex isotopic labeling-based quantification, and we are actually developing more labeling schemes with higher multiplexity in vivo, in vitro, and EPIQ for label-free quantification is under development. And because EPIQ achieves low LLOQ without demanding rare resources or demanding major change in existing experimental setup, we anticipate it can be adapted for diverse biological applications. And we are preparing zebrafish embryo sample for maternal zygotic transition time point study. So I'd like to thank my colleagues, Yeon Choi, sitting here, and Junwon Lee, and Dr. Jong Seo Kim, and my PI, Dr. Neri Kim. This work is funded by IBS. And if you're interested, please visit the post session P347 or email to me. And please don't be confused with another great poster session P449 for 2 EPIQ EPIC. Yes, there are 2 EPIQs in here. Okay, thank you. I will take the question.
I did not get how you then actually label the proteins.
What's that?
How do you label then the proteins? What kind of chemical modification do you use to differentiate?
Okay, so in LCMS, MS proteomics, proteins are digested into peptides.
Yeah.
So each peptide has N term and C term. In N term, the label is attached, and if the amino acid, the peptide ends with lysine, yeah, there will be attached label. So 2 possibilities. Yes.
Dimethod labeling?
Yeah, diethylation.
Diethylation.
Yes.
Okay.
Yeah. If you visit poster, you can see the detail.
Thanks.
Yeah.
I will do so.
Mm-hmm.